I'm going to be discussing cell communication. The first pathway is the G protein pathway. This pathway is the most commonly used pathway by cells. The pathway works with the help of a G protein. This is used to open transduction pathways and trigger cellular responses. The receptor protein acts as an on-off switch for the G protein pathway. When the ligand is absent, the G protein is held inactive by GDP, also known as guanine diphosphate. When the ligand is present and binds to the receptor protein, the receptor changes shape and activates the G protein by displacing GDP with GTP. This addition of phosphate is referred to as phosphorylation. The activated G protein then moves along the membrane and attaches to an enzyme. This changes the shape of the enzyme and activates it. The now activated enzyme triggers a cellular response. The G protein and activated enzyme are then dissociated and GTP returns to GDP, while the enzyme returns to being inactive. The G protein is now ready to be reused. The second pathway is the tyrosine kinase pathway. This pathway is mostly used for growth and emergency repair. Unlike the G protein pathway, which activates one protein, the tyrosine kinase pathway can activate 6 G proteins at one time. Before the ligands bind to the tyrosine kinase receptors, they exist as individual monomers. As the ligands bind to the receptors, the individual monomers come together to form a dimer. This process is known as dimerization. The two individual monomers coming together activates the tyrosine kinase region of each monomer and adds a phosphate molecule to each tyrosine cell. This occurs as ATP comes and ADP is released. Now that the phosphates are on each tyrosine cell, the activated receptor is recognized by certain relay proteins that bind to a specific tyrosine. This activates the relay protein by changing its shape. Each activated protein triggers a transduction pathway, which leads to a cellular response. The third pathway is the ion channel receptor pathway. This pathway uses a ligand-gated ion channel. This pathway acts as a gate by only allowing certain things to enter through the plasma membrane. The ligand-gated ion channel receptor remains closed until a ligand binds with the receptor. When the ligand is present, the receptor gate opens, allowing specific ions to travel through the gate. This rapidly changes the concentration of the cell, which can directly affect the way the cell functions. When the ligand disassociates from the receptor, the gate closes, shutting off all access the ions have to entering the cell. The fourth pathway is the intracellular receptor pathway. This pathway is mainly used for receiving hormones and steroids. For a signaling molecule or ligand to reach the receptor, it must pass through the target cell's plasma membrane. An example of a signaling molecule is a steroid hormone. Once the hormone has entered the cell, it will bind to an intracellular receptor in the cytoplasm, creating an activated hormone receptor complex. The hormone receptor complex will then enter into the nucleus and bind to a certain gene. The hormone receptor complex turns on genes by acting as a transcription factor. The gene is then transcribed into mRNA and a specific protein. Secondary messengers also play a vital role in cell communication. Secondary messengers are usually ion-based relay molecules in the cell's cytoplasm. The two most common types of secondary messengers are cyclin AMP, sometimes called CAMP, and calmodulin and inositol triphosphate, abbreviated IP3. When the ligand binds to the protein receptor, the GDP on the G protein is then displaced with GTP. The G protein attaches to the enzyme adenylyl cyclase. The enzyme catalyzes the conversion of ATP to CAMP. The CAMP, acting as a secondary messenger, then activates protein kinase A, leading to cellular responses. Thank you for watching, and that is the end of cell communication.